DNA replication is the process in which two identical DNA molecules are produced from one original parent DNA molecule. This process occurs in three phases: initiation, elongation, and termination. Here is a DNA molecule in the double helix. It is comprised of a sugar phosphate backbone and complementary nucleic base pairs. Adenine matches with thymine and guanine with cytosine. These are the enzymes involved in the three phases of DNA replication. In the first stage, initiation, enzyme DNA gyrase is ahead of the replication fork of the molecule and is relieving strain on the double helix, making it easier for enzyme DNA helicase to break down hydrogen bonds between bases, as we see here. Simultaneously, from the origin of replication, enzyme DNA helicase is moving along the double-stranded DNA molecule, breaking the bonds between the nucleic bases. This unwinds the double helix. Once unwound, only single strands of DNA are exposed, so SSBs, or single-stranded binding proteins, bind to the strands of DNA to prevent rejoining as well as damage. In the second phase of replication, elongation, enzyme DNA polymerase III attaches new nucleotides to the free 3-hydroxyl end of the strand and synthesizes in the 5-to-3 direction. Simultaneously, a primer is being created by enzyme primase. A primer is needed to carry out the synthesis of nucleotides. The lead DNA strand is synthesized in the same direction as the replication fork, while the lagging strand is assembled with multiple discontinued units called Okazaki fragments. Enzyme DNA ligase works to join these fragments together. This is elongation to the scale of a full replication bubble, as opposed to half a bubble shown previously. There are two lagging strands and two leading strands. The same synthesis is occurring on both sides. During DNA replication, there are multiple origins of replication, as well as multiple replication bubbles. These all carry out the same process and collectively synthesize new DNA strands, as shown here with DNA helicase breaking apart bonds between nucleic base pairs. In the final stage of replication termination, the new strands are complete and start to rewind into helical structure automatically. The RNA primer is removed from the 5 end of each daughter strand, causing it to be shorter. However, there are buffer zones called telomeres, whose erosions won't affect cell function. Enzyme telomerase is responsible for the extension of these telomeres, and DNA polymerase acts as an editor and removes incorrect bases.